So yeah, they'll do we pick up this question 2013, right? So because you have full question on molecular biology, we done certain things before, but I'm not sure whether you have confident enough to touch this kind of question, right? Probably you can get a lot of molecular biology uh, in this paper as well. So better to summarize this. Then I think I want to develop the confidence in yourself, in students, right? So question. Let me summarize the question. So what is the recombinant DNA technology? So it's a question. And then there's part A. Part B is describe the major steps involved in the production of recombinant, right? So the answer I show you, part B consists of actually 30, it's not ending here, another 15 following up, altogether 30 odd steps they are expecting from you, right? But probably if you're going to write steps, maybe you're not going to write according to the marking scheme. What do they expect from you, unless you don't have a understanding of it? So memorizing is not the solution, because memorizing doesn't serve the purpose. So that is why I picked this question. I want you to understand rather than memorizing. Okay. So just be patient. Right. So we go one by one. What is the recombinant DNA technology? Right. I go a little bit faster, but otherwise I can't finish this question. But please, I'm checking you whether you understood or not, time to time. So otherwise, you put your hand and let me to explain that, or you didn't understand, please tell me you didn't understand, right? So okay, because sometimes I don't realize students don't understand. So yeah, so you guys only have to tell me about it, okay? So Vidya, if you don't understand, please tell me, right? So okay, what is DNA
Mhm.
technology? What is DNA technology? For that part of the question is a small part. I just asking what is that. So then the definition, like recombinant itself, giving the meaning of the answer. Like it give the answer is hidden there. The recombinant DNA technology means two DNA molecules come from different species combined together. Right? That's part of the answer. So it is inside the question itself, right? So two DNA molecules come from not just different sources of course, but it it has to be from two different species. Otherwise, it won't become recombinant, right? So look at that. Joining DNA molecules obtained from different species. That is the first part of the answer. Second part is, so both together make a functional one DNA molecule. Both together make a functional, see, to make a functional single DNA molecule, right? So what do you mean by functional? So you can join two DNA molecules together from two different species. But if it is not functional, that is not the recombinant DNA technology. It has no technology actually, right? So the example, what we know for recombinant DNA technology is we already speak about the vector, right? It's insert and a vector. This insert, so DNA come from different source, maybe mammalian gene. This is a bacterial vector, all right? So this is how the very best example for gene cloning. We all the time talk about this vector plus insert. So this is good example for recombinant DNA technology, because once you insert into host, host is obviously bacteria because it's a bacterial plasmid. The host has its own chromosomal DNA, And this is plasmid is extra chromosomal DNA. Extra chromosomal means outside the main chromosome. So extra chromosomal DNA, so which has our insert, right? Our insert, and once you put it into the cell, it it can produce this. It can produce this introduced protein, right? Insert has an introduced protein which come from a mammalian or animal. So that protein is expressed, right? Or in other words, that protein is being produced inside the bacteria, like that. That protein molecule is inside the bacteria is now producing. If that happens, that is the recombinant DNA technology. So, to make the functional molecule, so functional means if you know it is functional or not, once you put it into the bacteria and see whether they can make protein, right? But we don't have to tell all of them. We just say functional DNA, right? Until it makes functional single molecule of DNA. So that's going to be enough for this answer for this question. This part of the question. All right. So is that clear, Devdu? So would you ask any
Yes,
doubt?
sir.
All right, all good.
Yes, sir.
All right. So we go to next part then. So next part, describe the major steps involved the production of recombinant bacterium. That now they're talking about. Previously they talk about recombinant DNA. Now they talk about recombinant bacteria that can produce useful protein. Now recombinant bacteria, in the sense, as example, we put that recombinant DNA into bacteria and let the bacteria to make the protein. Okay, so that is what this is. Now they are asking us in detail, stepwise, explain the process. That is not easy, right? Because you can lose lot of points because they give marks for the all the points. So if you lose the points, you lose the marks. If you're going to memorize that, maybe not appropriate, right? Because you might write something not matched into this question, right? So therefore, you need to understand. What I'm going to do here today, go one by one step, and explain you what does that mean in practical sense. All good. So first of all, identify the animal gene that produce useful protein. Having said that, they do now. So useful protein. So what kind of useful protein we can make? Right? Can we make all animal protein in bacteria or not? The animals are, for example, mammalian. We are mammalian too. So we have our DNA is in chromosomes. So we have eukaryotic system, right? We don't have plasmid. We don't have circular. All right, I just show you that. So this now we know that bacteria has a bacterial chromosome, which is entangled large. This is a large circular DNA, right? And this is extra chromosomal DNA. But this is a circular large DNA. But human being or other mammalians, they have different system though. So they have nuclear membrane, all right, and inside you have chromosomes. See? And DNA is surrounding or embedded in the chromosome. But this DNA is a linear DNA. So we have 43 chromosomes, right? 23 said 46 chromosomes. Every 46 chromosome has 46 linear DNA. So our system and their system is different. And in addition to that, we know in human DNA or mammalian DNA or eukaryotic DNA, you have what is called introns, and extrons. Remember? We talk about post transcriptional modification, right? The word is so big, but the meaning is what is transcription? And transcription is copying down to mRNA, right? Copying down to mRNA. Okay. If you forget, I remind that. So gene expression has two parts. First part is transcription. Other part is translation. Remember that? So transcription is copying it down to the mRNA. Translation is take this mRNA outside, right? So eukaryotic system only transcription. A prokaryotic system also has transcription and translation too, right? So ribosome attached to that and making protein, remember? So that is translation. So we talk about transcription. So transcription, once you make mRNA, it is transcription complete. But the mRNA we call is primary mRNA, right? Primary pre-mRNA or primary mRNA. Because this primary mRNA undergo transcriptional modifications, right? When transcriptional modification happen, one of them is splicing. What is the name? Splicing. So what is splicing mean? Yeah. So what is splicing mean? So we take this extron out. So introns are non-coding regions. Extrons are the coding regions. So we have different sections. After mRNA molecule get rid of the introns. So you got E1, E2, E3, right? So the thing is, the eukaryotic cell has freedom to join them in any particular order, right? Could be E1, E1, E1. It could be E1, E2, E1, E2, E1, E2. It could be any thousands of different choices. So therefore, one single fragment can make large number of different proteins. I'm pretty sure those sophisticated proteins cannot be processed in prokaryotic environment. So if you select a gene, so you need to select a suitable gene, or you could try it trial and error basis, whether it works or whether it not works. So not all the genes can express, right? So remember that one. Not all eukaryotic genes can express in prokaryotic vector or prokaryotic environment, right? But successfully done some proteins without, probably they are not involved in splicing. So two different genes. So some of them have splicing, some of them no need to have splicing, right? The whole thing, one extron itself is the protein.